Okay, guys, I am just going to introduce or just sort of explain a couple assignments that are due. Sorry, I'm trying to get this in the window here so that you guys can see. I think that should work. So I just have Moodle up right now just to show you where everything is. So basically, if you guys just took the exam, we're in this week here. If you look, there are a couple things. So one is when I have been meeting with you guys to talk about the lab report, I haven't looked at anyone's lab notebook. So what I want you to do is just take a picture of your cell culture lab notebook entry. It's probably a couple of pages, and submit it here. Okay, so I have the due date due by Monday. Please make sure you do that. Again, I told you that results, you don't really need to have anything in there except like maybe your raw cell counts when you wrote down those cell counts when you were actually taking them. That should be in your notebook. But in terms of right? So I think I kind of said at first that there doesn't really need to be any data in there, but you had to have written down your raw counts when you were looking in the microscope. That should be in the notebook under the results part, right? So that would be the only thing. You don't need to have like the graph of your data or any of the calculations done, but just those raw counts from those four squares that you counted should be in the results section, okay? And you should also have the methods in there. And then, a basic just little paragraph written about the conclusion. Okay, so make sure you upload a picture of that. This will be the last lab notebook check that I will do. Okay, so any of the other subsequent lab notebook checks that you might see on Moodle, we're not going to do. All right, so that's the first thing. The other thing is I want to introduce this Alzheimer's article. So you have another journal article summary done, due. Remember the rubric is up in a previous, you know what, I'll add it to here. The rubric is up in a previous week in Moodle, but what I will do is I'll also add the rubric here. So it is basically the same questions that I've been asking you all along. I do just ask you to choose one figure and explain what it's showing. So let me just introduce this kind of quickly and just sort of explain to you that this is a little complicated, okay? I'm aware of that. I just want to kind of just, again, sort of tell you why I chose it and sort of what to focus on, okay? So one thing, obviously this is related to Alzheimer's disease. It is looking at the effects of this drug, memantine, which is actually an approved treatment for Alzheimer's, and kind of just looking at the mechanism by which it might lead to behavioral, improvement, right? And so what it's particularly focusing on is phosphorylation, right? And so it talks about a phosphatase. And so the reason why I chose this article is because in the last chapter, we're talking about protein regulation, right? And that proteins can be phosphorylated and dephosphorylated, and that will affect their activity, okay? And so it's directly related to that. So I want you to sort of focus on that. The way that they're going to model Alzheimer's is by using basically a viral vector that will affect a gene, right? Gene expression, okay? And so they basically inject that viral vector into the brains of these rats, and that leads to Alzheimer's like an Alzheimer's like phenotype. Okay, hold on. Okay, so just to continue with that, and so again, remember that the model is induced. So the Alzheimer's model is basically caused by the injection of this viral vector. Okay, that again affects gene function. Okay. All right. So again, that's what the focus is. Okay. And so one of the figures that I particularly want you to pay attention to, or one that I think maybe is a little bit easier to interpret. Okay. Or there's two, right? Because you only need to choose one figure. So again, I don't want you to get so bogged down with the details here. The other reason why I picked this paper is that it has a lot of different techniques in it. And so one, this is what this is showing you here, right, is immunohistochemistry. So basically they took sections through the brain and then they're able to identify specific proteins with antibodies that are fluorescently tagged, right? And then you can actually visualize them. Now, if we were in class right now, we probably would have some slides that we could look at actually under our new microscope and actually look at some fluorescent labeling, okay? So that was another reason why I chose this as well. And so, like I said, one figure that I think I sort of think is important and that might be nice to sort of focus on is this one here. I probably can make this a little bit bigger. Oh, duh, right here. Okay, right? So if we look here, this is basically showing the effects of the memantine on tau hyperphosphorylation. So again, I mentioned at the beginning that this is about phosphorylation, right? And how it changes the activity of a protein. So in particular here, they're looking at tau, right? Which is a microtubule associated protein that is in neurons. And when it becomes hyperphosphorylated, it starts to aggregate together, right? And that's what forms those tangles that you see in Alzheimer's disease and also in other types of diseases as well. Okay, so these tangles form inside the cell and then it ends up causing cells to die. Okay, so what they're showing you is basically these antibodies, or so let's just say this, right? All of these sort of different panels are essentially different spots in the protein that are phosphorylated, okay? And these places in the protein are places that under normal circumstances shouldn't be phosphorylated, okay? So all of these, when you see an increase, suggests that they're hyperphosphorylated, okay? And what I want you to particularly pay attention to is the blue and the purple here, right? So the blue is basically their model, right, where they inject this viral vector, that it essentially increases the amount of phosphorylation, okay? And then again, that sort of leads to this sort of Alzheimer's model. And so you can see that, again, if you look at pretty much all of these panels here, and you look at the different brain areas, right? That's what they're looking at down here, different brain areas. You can see that that blue bar is always higher, right? It's significantly higher, meaning there's an increase in phosphorylation, okay? However, if you look at those same animals, that Alzheimer's model, right? When memantine is given, you can see that it's decreased. Okay, so it's saying, and what they're showing is that the memantine is decreasing the amount of phosphorylation. Okay, and then again, these are controls, right? Well, so the GFP, again, if we look at this picture that I showed you here, right? GFP is just this green fluorescent protein. So it's just the control. And it's a way of identifying, it's basically injecting the viral vector without anything in it, right? So it's not going to change any gene expression or anything like that, all right? And so you can see that that's the sort of, it's these GFP ones are normal rats. Think of them as normal rats injected with the memantine, and then normal rats injected with vehicle, which means like saline or something like that. Okay, so like I said, the what to pay most attention is this. Now, this is now showing you up here our Western blots. Okay, so don't worry about that. I would just focus on this because it's easier to visualize what's going on here. And so these are the actual blots to which this is quantifying. Okay, so again, each one of these it's a different antibody that recognizes a different place where the phosphorylation is occurring. Okay, all of which it's sort of abnormal to have this increase in phosphorylation. All right, so again, this is the Western blot that then the actual blot to show you, but it's quantified down here. Okay, so again, paying most attention to here. If you're going to describe a figure, this is probably one that might be a little bit easier to describe. Also, another one might be one of the ones where they focus on the behavior. Okay, so there's a lot of data in here and a lot of figures. Like I said, don't get bogged down in the details. But this might be also one that you might want to describe. And so basically panel E is probably the one to pay most attention to. And again, you have the same groups here. And basically this is a task where it's sort of a learning and memory task. Okay. So what you see is that these guys, right, in blue are the Alzheimer's model, right? And you can see that it takes them, they travel a lot. So bottom line is that if this number is higher, this is the distance it takes them to travel to find a hidden platform, okay? So animals that know where that hidden platform is, right? They've learned the place of where it is using, spatial memory or using navigation sorts of strategies. They know where that platform is. Therefore, when you place them in this water maze, they quickly go to the platform. okay? However, if an animal doesn't know where that platform is because there is some sort of disturbance in learning and memory, right? Because again, at day 3, they've been exposed to this a couple times, they will still take a long time because they're going to be searching around the maze to look for that platform, okay? So again, what you see here, now this is showing you functionally or behaviorally, you see that the vehicle animals, right, the ones that didn't receive the drug, but they're the Alzheimer's model, they're still taking a long time to try to find that platform. However, if you look at that Alzheimer's model, right, that's been treated with the memantine, you can see that they look like the control animals, okay? So again, those are 2 figures that I would focus on and sort of describe in your own words when doing this journal article summary. Okay, so I hope that helps. The other thing I want to mention to you guys, and so that's due Friday, April 17th. Your final lab report is also due Friday, April 17th, so that's next Friday. Well, I shouldn't say next Friday because I don't know when you're listening to it, but it's Friday, April 17th. So let me go back. Okay, so that's here, this Alzheimer's article summary. As I said, the lab report final draft is also due on April 17th. You're going to be submitting it. If you look here, it looks a little different. This is a Turnitin assignment where basically it checks against published literature, and it also checks against other people's work in your class to make sure that there is no copying or plagiarism going on. So when you're citing a source in that final lab report, and I've kind of talked about this with everyone in their individual meetings, you should just be paraphrasing that, right? And then citing it properly, okay? You shouldn't be taking things word for word from that paper. And if you do, the percentage of things that are the same between published literature is going to be very high, okay? And that's an issue. Obviously, when you're writing scientifically, we can't copy things. We cite it, but we don't use quotation marks, right? It's just basically you're saying I'm taking or I'm getting this idea from this paper, but I'm not copying their exact words, okay? So anyway, that's due the 17th as well. The other thing is this PCR lab assignment, okay? So we haven't talked about that. Okay, so as I said, the other thing that is due next week is this PCR lab assignment. And that is due Wednesday, okay? Just so that we can talk about it on Wednesday if necessary. And so there, I'll show you, there's this PCR student lab manual, okay? So that's in the folder there. And basically the assignment is to read pages 30 to 33, answer the questions on page 34, then read 35 to 36 and answer the questions on page 37, type up those answers, and then submit them here, okay? So like I said, this is due Wednesday by 10 a.m. by class time. All right. So I just wanted to introduce the PCR procedure because, again, you know, we would have been doing this in class, but we're not actually able to do it. So I always sort of go over the, you know, the background information about it, and then you guys would have actually been able to actually do a PCR, okay? So here, oh, why is it doing that? I'm not really sure. Oh, I think it's because of the layout. Oy, oy, oy. Crap. Let me just see if I can figure it out really quick. It's not the slide layout, it's the way the slides are. Hold on a second. Okay, so again, just to give some background information on this PCR lab. Okay, so we all remember DNA replication, right? Hopefully. And, you know, some sort of the main enzyme that's involved in DNA replication is DNA polymerase, right? And they have 2 fundamental properties. They can synthesize DNA in that 5' to 3' direction, and they basically add new DNTPs to a primer strand, right? And so there has to be this primer strand, a primer there first, and then basically you can add those nucleotides to that primer strand, okay? And so again, you guys saw this picture before. This was in our lecture on DNA replication, but here's the primer, right? And then the polymerase will add these nucleotides in the 5' to 3' direction. based on complementary base pairing. Okay, so again, the reason why we're talking about DNA replication is that PCR is basically based on DNA replication. Okay, ooh, that's not very good. So let's fix that. Sorry, I had to change the layout and it messed with my slides. Okay, so it says detection of nucleic acids and proteins. So it says so let's kind of start this way. So in order to be able to study DNA, right, nucleic acids, you have to be able to have a lot of it, right? And so if you just took a piece of your hair, or you took blood, or you took some cheek cells from your cheek and you extracted DNA, you have DNA there, but it's probably not enough for you to just study, right? And if you think about like a crime scene, because that's sort of the assignment is based on this sort of this crime scene DNA lab that we've done in the past, you wouldn't be able to analyze that unless you could make more of it, okay? And particularly with PCR, you're going to make more of a certain region that you're interested in, okay, by using a primer. Okay, so it says to isolate large amounts of single DNA molecule, you don't need to do molecular cloning. We're not going to talk about that, but that's a much more complicated process. You can use PCR. And so, like I said, you could extract DNA from hair, from saliva, from wherever, right? And then basically you can take that DNA, add it to a tube, add, polymerase and all the stuff that needs to happen for replication, which we're going to talk about in a second in terms of components, along with a primer. And then you can amplify a certain region of that DNA. Then you can study it, okay? And so this PCR, polymerase chain reaction, was developed in 1988. So it's a pretty recent technique. So it says DNA polymerase is used in vitro, right? Meaning in a tube, like I said, to basically do repeated replications of a defined segment of DNA, like I said. So that's just saying it in different words. Apparently I'm going to have to fix these every time. Okay. So again, I think I said some of this stuff already, but just to sort of reiterate. So a specific region of DNA can be amplified if that nucleotide sequence surrounding the region is known. So basically you design primers, or if you're interested in a certain gene, you're going to know what primers you want to add to your tube, right? So you have to sort of know the sequence. There's many ways where you can do this. You can design your own primers. You can buy them commercially to basically target a specific gene. But there are many, many resources to design those primers. But bottom line is you have to know what you're targeting, okay? So you basically have two primers that are designed either by you or by somebody else to initiate DNA synthesis in both directions, okay? So you're going to have one primer on one strand and another primer on another strand. And they're usually about 15 to 20 bases long. Okay, so that's one thing you need. You need those primers, right? Now, the PCR reaction is going to start by heating the DNA template to about 95 degrees to basically separate the strands. So if you remember in DNA replication, you need to separate those strands and sort of unzip that DNA, right? The DNA polymerase then has access to each strand and basically can, right, add those nucleotides based on complementary base pairing to that primer that you added. Now, there's a specific special polymerase that's used in PCR, and it's called Taq polymerase. And the reason why the only polymerase you can use to do this procedure is Taq is because it's heat stable. So this polymerase is actually derived from a bacteria that lives in hot springs, right? So obviously super hot, and therefore its enzymes, particularly the Taq polymerase, have been adapted to be able to survive those high temperatures. Okay, so Thermus aquaticus is the species of bacteria to which this Taq polymerase has been extracted from. Okay, if you used a regular DNA polymerase like a human DNA polymerase and then you basically heated it to 95 degrees, I'm sure you're thinking exactly what would happen to it. It would be denatured and it would not function. Okay, so this heat stable polymerase, this Taq polymerase, is a key component to being able to do PCR, right? Or essentially DNA replication in a tube. Okay, because the only way you can get those strands to unwind is by heating them. Okay, and basically you're going to keep doing this repeatedly, as you'll see. Okay, so you're basically going to repeat what I just said over and over again. okay? So it's basically the PCR reaction is a series of temperature changes. So you put all the components in the tube, including your primers, right, your polymerase, all the things that you need, free nucleotides. So the polymerase has access to those free nucleotides and can add them. And then you change the temperature, okay? And so temperature is heated to 95 degrees, then it's lowered to allow the primers to pair, right, and to adhere to the DNA, and then to allow DNA synthesis to proceed, okay? And so basically that's the replication part of it is happening now once that temperature is lowered. I'm going to show you a picture. And then basically you repeat that cycle multiple times, okay? Each cycle you will have a twofold increase in your DNA, okay? And so this can amplify the DNA rather quickly. And so here's your starting DNA, and you heat the strands to 95 degrees, separates the strands. You lower that temperature to about 55 degrees, 60 degrees, depending, and that allows those primers to bind. And then that Taq polymerase comes along and adds those nucleotides here and basically makes more of this DNA, right? And so again, you end up with 2 new strands, right? You started with 1, now you got 2. Each cycle, you get 2 more, okay? And so each cycle, you have a doubling of that DNA. And so again, it amplifies that DNA. And again, it's only amplifying this region here between those 2 primers. It's not replicating the entire genome, okay? So this, and that's important. It wouldn't be useful if you're replicating the whole genome. You want to be able to target a specific region, okay? And then again, once you've amplified this specific region of DNA, there are then many things that you can do with it. One of them being, analyzing it for, forensics, okay? And that's sort of the application that we're using and what your assignment is tied to. So I've said all these things all along in terms of what needs to go in that reaction mixture: primers, right? Taq polymerase, again, nucleotides, okay? And again, each cycle doubles the amount of DNA. I said all that already, okay? So as I was sort of saying through the presentation is that our sort of application here is sort of this crime scene investigators, okay? And in this, you are amplifying a specific region, okay? So it says here, so DNA sequences used in forensics are non-coding regions. And again, the background information and the questions you're going to do will relate to some of this, okay? Non-coding regions that contain short tandem repeats, okay? So they're called these STRs. And so this is the locus and it's on chromosome 11. So there are primers that specifically target this TH01 locus, okay? And it's because there are many differences from person to person in this region. So by looking at those repeats in that region, between person 1 and person 2, they will be different. There are many, many regions of the DNA that are pretty much the same between individuals. Okay, so it's important in forensics that they're picking these particular regions that really vary from individual to individual. Okay, and so, like I said, those primers will be specific to that locus. Okay, and again, you guys are going to read through that lab packet and answer those questions like I showed you before. Okay, so that was just a little bit of background information about PCR in general, and then the actual assignment is here, right? So PCR lab assignment, again, is to read those certain pages and answer those questions. The lab packet is in here. That PowerPoint that I just showed you is in here as well. Okay, so if I click on it, you can see that here. All right, and so I think I went over everything in terms of what's due in basically next week, right? Remember, we're on Easter break. I am continuing to meet with you guys for your individual, for your first draft of your lab report. But then now these are the assignments that are upcoming for next week. So Alzheimer's article summary, we went over that. I introduced that article to you. Picture of your cell culture, lab notebook entry, and this PCR assignment, okay? Also, that lab report final draft is also due next Friday, okay? In addition, we will meet next week, right? And I'll put that up and I'll send you guys a reminder about that, but we will be meeting on Monday and Wednesday next week. And on Monday, we can sort of discuss and review the cytoskeleton a little bit. And so the recordings are already here, okay? And as you can see, there's a quiz that's tied to this. So the next topic for you to be reviewing is cytoskeleton. Okay, so cytoskeleton lecture 1 and lecture 2. And then from there, we'll talk a little bit more about the plasma membrane, particularly membrane potential, sodium potassium pump, and then move on to cell signaling and cell cycle. And I will then upload recordings of that. But again, another thing to do, cytoskeleton quiz is due by Wednesday. Okay, so you have quite a few things kind of, up and coming. But again, we'll spend next week sort of reviewing cytoskeleton and maybe starting to talk about plasma membrane. I will have those recordings up as well. Okay, so I think that is pretty much it. If you have any questions, just let me know. Thanks a lot. Bye, guys.